I would like to acknowledge my support and my financial disclosures. And the question I want to raise today is, what is the front-end biology of AMD? And by analogy, let me mention cardiovascular disease, where elevated plasma LDL, the bad cholesterol, is a major known risk factor. But LDL is also part of a system to deliver fuel, lipid, and lipophilic vitamins to the body cells. So we actually want LDL in our body. If you consider cancer, cell division would be considered dangerous, but cell division is also necessary for normal development and wound repair. So we actually want cell division to go on in our bodies. So the question I want to address is there a process that originally evolved for good vision, and did that become maladaptive and lead to AMD? And I thank the preceding speakers for laying a nice groundwork on that. So from that, I will say that my bottom line is that it's time to rethink the cellular basis of macular xanthophyll pigment. The Mueller glia in the retina are the major repository of the xanthophylls. The xanthophil delivery to Mueller glia is a plausible explanation for the abundance of dietary delivered fatty acids in soft drusen, which are the major intraocular risk factor for AMD progression. The evolution of neuroglial relations underlying human foveal vision came with a price, the development of soft drusen and their sequela long after our reproductive years. So this is a figure we've already seen. It's one of the most famous images in vision science, and that is the localization of macular pigment in the fovea of a monkey, shown by Max Snodderly and Francois Delory over 30 years ago. What they described was a localization in both the inner and outer retina, describing a main peak of absorption in the region of the receptor axons and a second strong but smaller peak that follows the inner plexiform layer. So those are that's the outer retina and the inner retina. And over the years, this has evolved into the understanding of many people that the macular pigment is localized only in the cone axons. And it's time to correct that misconception, because the Mueller cells are the radial glia of the retina. They span the retina from the inner limiting to the outer limiting membrane, and that they're distinctly Z-shaped in the macula. In this schematic, the orange spots are the nuclei of the Mueller cells, and now you can see the Z-shaped cell. These Mueller cells contain specific markers shown over here on the right. If you look at the population of cells in the Henle fiber layer, where all those oblique fibers are located, you can see the cones, which start with the inner and outer segments down below and end at the synaptic terminal up above. You can see the rods, which actually outnumber the cones in human macula, and they follow the same course. And the Mueller cells are interleaved between the rods and the cones, and their cell body is in the internuclear layer, and they traverse the whole retina. So these, of all the retinal cells, are the most capable of accounting for the distribution of the macular pigment. So in addition to the distribution that matches the masked morphology of individual cells, there are a number of other lines of evidence, primarily from the MacTel study, which has shown both histologically and in imaging the loss of macular pigment in eyes with macular telangiectasia 2. You can see that here. The control eye has a Mueller cell marker that's absent in the diseased eye. By fundus autofluorescence, you can see the loss of screening by macular pigment in the same area where the cystic degeneration in the fovea. And in surgical cases, it's possible to excise epiretinal proliferation, and the excised tissue contains Mueller cell markers and yellow xanthophyll pigment. You can see that there. So there's a good reason to believe that the Mueller cells the Mueller glia are actually the major repository of the macular pigment. So we decided to get involved in this field by way of transmission electron microscopy, which is a comprehensive visualization technique for neuroglial interactions. And excellent preservation is actually possible in human tissue post-mortem, as you can see by this specimen. And furthermore, 3D reconstruction of cells is now accelerated with new alignment technology. And our goal was to eventually visualize an entire Mueller cell, which has never been done before in human macula, and then to quantify the abundance of Mueller cell biomass in macular tissue. So we approached this the foveal connectome is the brainchild of Dennis Dacey at University of Washington. Through 4,065 nanometer thick sections, you can see this cube there of 187 identified cones in the center of the fovea. Cone 1 is the red one right there. And we targeted those for reconstruction. The color map on the left shows the 187 cones, and there's a lot of space in between them that's occupied by Mueller cells. And in fact, over 40% of the surface area of the retina is occupied by Mueller cell tissue. So they're really an important part of the foveal structure. Here is a 3D reconstruction of cells, neurons in the internuclear layer. There are two different bipolar cell types with a Mueller cell in between, right there. The numbers refer to the catalog of reconstructed cells. So this is a bipolar sandwich. So to quantify the biomass, you can see by the cartoon over on the left, the light shines in above through the vitreous, and the sclera is at the bottom of the slide. We're going to peel out this bipolar cell and look at the associated Mueller cell area. There's high Mueller cell cross-sectional area in the two synaptic layers shown at the top and the bottom of the graph. And you can see that here in the 3D reconstruction, how the shape of the cell changes, and you can appreciate this in what I hope that is. The video is not working, but if it did, it would show you how the cross-sectional area changes across the different length of the Mueller cells. So there's also evidence that the Mueller cells, the Mueller glia in the center of the macula differ on a molecular basis from the Mueller cells in the rest of the retina. And this is our recently published results in both monkey and human, showing a toluidine blue staining of a population of cells that delimits the fovea above the yellow arrowheads, and you're going to see a similar kind of configuration in a human as well. There is, and we don't know what the basis of this differential staining is. We just know that it tells us that there's a population of molecularly distinct cells in the center of the human fovea. So we've recently approached this with imaging mass spectrometry, which is a very powerful technique. It's a label-free imaging method that produces images based on molecular weights of tissue components without using any antibodies or stains. So IMS allows imaging of lipids, proteins, peptides, other molecules in tissues. And you can see how it works. There's a flat, up above, the flat-mounted retina right here is on a slide. A matrix is applied. Laser shots are hit across that tissue in a systematic manner, and the ions fly back to the detector, and it's possible to map them all. And here is a map of a prominent retinoid derivative called A2E. Now, it's possible to do this also in tissue cross-sections. There's a workflow described up above and illustrated down below. In tissue cross-sections, They're first viewed with with fluorescence microscopy to see the layers. Then the MALDI IMS show the layers of molecularly distinct mass over charge. There's an autofluorescence post-acquisition of IMS, and in this corner right here, you can see the grid of laser shots. Sorry, advanced. And then finally, the sections can be stained with H and E to show the tissue layers. And to go back to that classic image of the xanthophyll distribution in human macula, that's a monkey. Here, this is a human macula, and you can see the same configuration. And this is a distinct mass over charge associated with 1-phosphatidylethynolamine. We can also see, and this, let me just back up and say that this particular signal may or may not have something to do with the macular pigment, but we do think it has something to do with the Mueller cells. So it's probably in the same cells that are harboring the xanthophylls. This is another a mass over charge for which we do not know what the molecule is. It's probably localized to the Mueller cells plus because they seem to radiate far out from the foveal center, but it's also detectable in the RPE cells that are outside the macula, which makes sense in terms of what we know about visual cycle processing in the retina. And here's another example in which the signal is localized only to the fovea. So, having told you all of that, what is the connection to AMD? Okay, so I'm very focused on the deposits in AMD, drusen in particular. These are the characteristic extracellular deposits, and our model is that soft drusen are a downstream consequence to the constitutive dietary delivery of lipophilic essentials to macular cells, and then the RPE-mediated recycling of unneeded lipids from these sources out to the circulation. And I want to focus on this one phrase in here, which is the constitutive dietary delivery of lipophilic essentials. So you should see where I'm going with that. This is a diagram of the outer retina. To put it in context relative to what I just showed you before on the fovea would be off the top of the slide up by the ceiling. So this is a greatly magnified view of just the photoreceptor tips, the RPE, Brooks membrane, which is a vessel wall behind them, and the choriocapillaris, which is the capillary bed. And you can see some of the players, and you can see the circle over on the left, which shows some of the lipophilic essentials that are delivered by diet, including cholesterol, lutein, and vitamins E and A. So how do we get from AMD back to that biology? So drusen abundance is the largest intraocular risk factor for AMD progression. It's been shown over and over in multiple studies. And now the pathway from drusen to both neovascularization and geographic atrophy, which are the two end stages, are known from clinical imaging. I'll show an example of that. Soft drusen can be modeled as masses of lipoproteins that are trapped by, secreted by the RPE and trapped by aged Brooks membrane choriocapillaris complex. Brooks membrane lipoprotein composition implicates diet and outer segments as the two major sources. And then the HDL-mediated delivery of xanthophyll pigment is a plausible upstream dietary source that would account for the fatty acids in drusen. So I'm going to step through some of these things one at a time. And here's a diagram then to show the, a little bit bigger magnification, the tips of the outer segments on the top, the retinal pigment epithelium, the cell layer in the middle, and you can see a drusen there made up of the masked lipoproteins, and these are localized between the basal lamina of the RPE and the inner collagenous layer of Brooks membrane. So the fundamental work on drusen heterogeneity, ultrastructure, and clinical significance was done by Shirley and John Sarks, who are two ophthalmologists active in Australia, and their electron microscopist, Murray Killingsworth. And you can see the soft drusen there and the hard drusen. And since their descriptions 20 to 30 years ago, we've learned through clinical imaging that soft drusen are dynamic with a life cycle that's observable in vivo. And I'm going to show you some examples of that. Let me skip ahead here. On the right-hand panel is a series of spectral domain OCT images. These are scans through an individual patient over the time frame indicated. The RPE elevation right here this is a big drusenoid pigment epithelial detachment. The yellow arrows are hyperreflective foci, which are RPE cells that leave the drus and migrate into the retina. Leaving these streaks called hypertransmission, which indicates the atrophy of the RPE layer. And this can all be quantified, and that's shown over here in the other panel. Okay. You can see the growth of drusen and the collapse. So they grow slowly and they collapse. We quantified this for larger drusen, and it also happens for small ones. So to elucidate our model of soft drus biogenesis, the soft drusen are very lipid-rich, and that's because the RPE is a native secretor of ApoB and E containing lipoproteins that combine lipids from photoreceptors and diet. So as described by the SARCs, the principal component of soft drusen and basal linear deposit are membranous debris, and you can see that in this magnified electron micrograph. It does look like membranes. But if you use appropriate lipid preservation techniques, you can see that what looks like membranous vesicles in conventional EM are actually solid space-filling lipoprotein-like particles by quick freeze deep etch. So these lipoprotein particles accumulate with age in Bruch's membrane, especially in the macula. The particles fill in towards the RPE. They form a layer on the Bruch's membrane inner surface, which we call the oil spill. And that's the transition between aging and AMD. So we also determined that aging Bruch's membrane accumulates esterified cholesterol. This shown here with oil red O binding and Philippin staining for esterified cholesterol. This is a large universal constitutive process in normal retina. It's 7 times greater in macula than in periphery. 60% of cholesterol is esterified. It's been multiply confirmed. And the fact that it's extracellular esterified cholesterol implies a lipoprotein because that's the only way you can get esterified cholesterol outside of a cell. This is a very busy slide. To skip the graph and just look at the header. In a microarray study of human retina and RPE, we found evidence for expression of 84 genes involved in cholesterol and lipoprotein handling. These were well known from atherosclerosis research, and that's why we tested in the retina. And it's also been confirmed by RNA sequencing. And importantly, the retinal pigment epithelium expresses ApoB, which is the backbone of plasma LDL, and microsomal triglyceride transfer protein. So the RPE is a native lipoprotein secretor, just like liver and intestine. Okay, so the composition implicates diet as an upstream source. So lipoprotein lipids are assembled from many sources, and you can see that the Bruch's membrane lipoproteins, they're isolated. The image on the left shows that. They're very esterified cholesterol rich despite their large size. So they're unlike plasma VLDL that way. If you look at the fatty acids that are esterified to the cholesterol, they're highly enriched in linoleate, just like the plasma lipoproteins. And they have hardly any docosahexaenoate, DHA, which is the signature fatty acid of outer segments and all neural tissue. So from this, we deduced that the fatty acids were derived from dietary sources. It's been repeated in 4 labs with 5 techniques. One of them was a study by Paul Bernstein. So to summarize this model then, we believe that there's uptake of plasma lipoproteins delivering xanthophylls and vitamin A that comes in by the LDL receptor and scavenger receptor by the RPE, and it's combined with the lipids from shed outer segments, and the excess fatty acids and cholesterol are recycled via ApoB and E-containing lipoproteins out to Brooks membrane. It's retained in aging Brooks membrane, becomes pro-inflammatory, pro-angiogenic, and that's how you get type 1 neovascularization. So we've gotten experimental support for this diet-driven model in that RPE in culture that's well differentiated and polarized can lay down a layer of deposits, fed only the culture medium. So they make a continuous layer of deposits, some like drusen here, depending on the surface that they're grown on. So this shows that diet is indeed important, and outer segments are actually not required for the process. And we take from this and the clinical imaging that soft drusen form because the RPE is functional and the clearance through Brooks membrane and choriocapillaris is impaired. So to go back to the neurosensory retina and to reconsider where does macular pigment fit in all of this, we can use topography of the macula to dissect mechanisms. This shows the topography of rods, cones, and RPE cells in normal human eyes. The macula is delimited by the dashed lines there. The red line indicates the peak of high cone density. The green line indicates where rods are located and how they fall to zero in the fovea. And then the brown line shows the RPE has a slight rise in the foveal center. Now, if we go and look at that in more detail, here I've calculated, I just noticed that the x-axis is wrong. These are ratios. So what's plotted here is the ratio of photoreceptors to RPE. Here is the limits of the macula. This is the area in which rods tend to be lost in aging. It's a very characteristic effect in aging. And you can see here that the ratio of photoreceptors to RPE is actually lower in the macula than it is elsewhere. So the idea that phagocytic load of RPE is important, it may be less important than we think. Here, underneath all of the retina is the accumulation of basal linear deposit and soft drusen, which we mapped and published several years ago, and it's mapped up against macular pigment. And you can see that the macular pigment is vertically located above, and it makes sense that these very lipid-rich processes are related to each other. So this is a complex diagram, and I'm just going to point out once again lutein and zeaxanthin delivery, and how does this cross over to the retina. Recent work by Paul Bernstein implicates IRBP. There are other mechanisms, perhaps an intraretinal HDL based on ApoE, analogous to what's available in the brain. So all of this is speculative, but if it's correct, it could explain foveal sparing in AMD because that's just another example of Mueller cells caring for foveal cones. It could explain recurring reports of high HDL as a risk factor for AMD, and that's because there may be a mass effect of HDL bringing in xanthophylls, resulting in high uptake, but also a need to offload more lipids at the end. It suggests that isolating brain glia to study xanthophyll pigment role in cognitive health might be a prioritized research direction. Maybe people are doing this already. A concept I learned from a Jim Stringham paper was neural efficiency, that this hypothesis of what xanthophylls are doing could be prioritized for future study, and this is something that Paul's animal models might be useful for. And then it connects, I think, the end stages of AMD to the evolutionary biology that gave humans foveal vision. I've spent 25 years learning about AMD from cardiovascular disease, and I'm going to summarize a whole lot of details by saying if we start at the clinical end stages and complications of neovascularization and calcification, we work back to cell, tissue, and molecular mechanisms. And for AMD, it's an atherosclerosis-like progression in a vessel wall, which is Brooks membrane with an interocular lipoprotein source. And then the evolutionary biology, which led to all this, was lipid delivery. So to conclude then, it is time to rethink the cellular basis of macular xanthophyll pigment as the Mueller glia, as the major repository in retina. Xanthophyll delivery to Mueller glia is a plausible explanation for all the linoleate-rich fatty acids in soft drusen, which are the major interocular risk factor, and the evolutionary of neuroglial relations underlying human foveal vision came with a price, and that's the soft drusen and the sequela of soft drusen long after reproductive years. I want to thank my many co-authors over the years. Thank you for your attention. This is all in a review that will come out in IOVS very shortly. Thank you.
We're happy to take questions.
Any questions?
Any questions from the floor? Yeah, we've got.
Thank you very much for a very interesting presentation. Could I ask, where do you feel that the defective complement inflammation and macrophage-driven inflammation story in the pathogenesis of AMD fits in with your model?
That's a really good question, and I knew somebody would ask me. I would like to have an answer to that, except that the full range of cells in the retina and the choroid that express complement have not been delineated. We actually don't know where all of that occurs. There's a very strong signal for CFH, for example. It's in endothelial cells of the choroid. There's also strong signals in retinal cells, individual cell classes. So I would say that we don't really know. One of the strongest indicators for a role of complement was the presence of complement-related proteins in drusen. They were all, the studies that did that used peripheral drusen. Okay, it's not clear that they used macular drusen. In fact, there are two published reports that did not show complement-related proteins in macular soft drusen. So I think there are a lot of open questions about what complement is doing, and I firmly believe it'll hook up to this somewhere, but that's still really an important open question.
Thank you.
There you go. I'll go back again.
Thank you, Christine. That's really interesting work. I think I may have missed this, but can you speculate as to why you think that the Mueller glial cells in the macula might be so specialized to retain the macular pigment, as Mueller glial cells are distributed throughout the entire retina?
No.
Well, that's the $64 question, right? Why do we have a fovea? Why are the cones concentrated in the center of the macula? I think it's part of the same package, that it's a product of evolution for excellent vision. And
well, but certainly, I mean, the cones are distributed in a way that they're accumulated more in the macula and fovea, of course, but the Mueller glial cells are distributed.
They're everywhere. Well, cones are across the retina too.
Of course, but in much less density in peripheral regions.
So one of the reasons is that the Mueller glia perform many services for cones. And one of them, which has been explored essentially not at all in primate or human retina, is the delivery of vitamin A. So there's a second visual cycle. I mean, the visual cycle that's well known is the one that involves the RPE, delivering 11 cis retinal to photoreceptors. the second visual cycle involves the Mueller cells delivering 11-cis retinol to cones. And that was first described in early 2000s by Nathan Mata and Gabe Travis, and more recently by Vladimir Kefalov. And that has not been explored exclusively, this has not been explored in primate retina. So, but if this is all true, then you need a system in place in the macula to supply vitamin A derivatives to the cones. And that's probably probably the major driver of this whole evolutionary process, is providing the chromophore to the cones. And the xanthophylls are part of the package. It's one that we can see, it's one that we can visualize, so we can actually study it.
Okay. We have one more question up front.
That was a great talk. I like the analogy with atherosclerosis, because it's the first thing I understood this morning. But my question is, I think I missed you. Are you saying that the secretion of the ApoB-containing lipoprotein is sort of driven by the necessity of moving a lot of xanthophyll out of the cell? Did I miss that?
So this is a multi-step process. So xanthophyll and vitamin E and everything else that comes in from diet is delivered. The RPE takes out what is needed by the photoreceptors and sends it on its way, and then it has all this excess lipid to get rid of. It's not needed. And so the ApoB system is a way to get rid of that.
But there's a lot more cholesterol to get rid of, probably, than there is of xanthophylls. The drusen are very cholesterol rich. The lipoproteins in Brooks membrane are very cholesterol rich. And that, we think, the cholesterol is harder to trace because it's it doesn't have a fatty, but we think that's coming from the outer segments. Now, outer segments are actually relatively cholesterol poor relative to other membranes, but the fact that the RPE takes that in every single day, that in bulk it has a lot to get rid of. So it's really dual source: cholesterol from outer segments, fatty acids from the leftovers from diet, and that's what comes out as drusen. And as Brooks membrane and the choriocapillaris complex essentially stiffen up and become less efficient with age, it piles up there, and that's what makes the druse. Thank you.
So we're running a little behind schedule.
Yeah, sorry.
Thank you so much, Dr. Curcio.